Today's video is about protein sequencing. We will discuss what is the definition of protein sequencing, which methods are used in protein sequencing, how it is done, why it is done, and what are the challenges in protein sequencing, or why protein sequencing is not as common as DNA sequencing. So let's start the video. Protein sequencing means determining the amino acid sequence of part of protein or peptide. The methods which we mostly used in protein sequencing are three methods. These are Edman degradation, tandem mass spectrometry, and bioinformatics. These methods are mostly used in protein sequencing. The steps which we use in protein sequencing are the following. First of all, we split or break down the polypeptide chain. After breaking the polypeptide chain, we determine the number of polypeptides that are present in molecule of protein. And after determining the number of polypeptides, next step is break down the disulfide bonds that are present between amino acid molecules. The other or next step is detecting the amino acid composition of polypeptide chain. After that, we sequence the two ends of a protein that are amino terminus or carboxyl terminus. These two ends of protein are sequenced in that step, and after that, sequencing the ends of protein, then we cleave or break down the polypeptide into smaller peptides. Now we have more fragments, short fragments of number of amino acids combined, and then we determine the sequence of amino acids of each peptide, each fragment. After that, we determine the sequence of peptide fragments. Now, sequence of peptide fragments means that in which order, in which arrangement, different peptides are combined to form a complete molecule of protein. The last step is identification of position of disulfide bonds in original polypeptide chain. In that step, we see that where in polypeptide chain of protein disulfide bonds are present. The next section is why protein sequencing is done. What is the importance of protein sequencing? Protein sequencing can be done for drug industry and for antibody testing, and some other important points are here. Protein sequencing generates significantly fewer random matches against sequence libraries than DNA sequencing. As compared to DNA sequencing, if you talk about protein sequencing, there are fewer random matches, less confusion, more accuracy in this method. The next point is that redundant sequences, which are meaningless sequences or non-functional sequences, are not very common in protein sequences as compared to DNA databases. DNA databases have more redundant sequences as compared to protein databases, which makes it more accurate, more easy to sequence a protein. DNA databases are more expensive, they are more costly as compared to protein databases. The next point is that protein sequencing provides information on post translational modifications. Now, translation is a process in which protein is formed at ribosome from messenger RNA, and there are certain modifications in protein which are called post translational modifications. These post translational modifications are important, and their understanding is especially important for understanding or for finding the causes and treatment of different diseases. Now, the last section of today's video is that what are the challenges in protein sequencing? Why protein sequencing is not as common as DNA sequencing is? DNA sequencing is rapid and laborious. It is rapid and less laborious. It means less effort is required in DNA sequencing as compared to protein sequencing. Now, the next 3 points will justify why DNA sequencing is more rapid or less difficult. DNA sequencing is rapid because DNA has only 4 nucleotides. DNA has only 4 nucleotides, so which are easy to sequence as compared to protein, which has 20 amino acids. So the number of units which are involved in the composition of two molecules are different and more in case of proteins. So that is the reason that protein sequencing takes more time. Next step is that DNA is charged molecule. Now DNA has a negative charge due to presence of phosphate, and because of being a charged molecule, DNA molecule undergoes ionization more easily as compared to proteins. And so this ionization helps in mass spectrometry. Therefore, DNA sequencing is easier as compared to protein sequencing. In DNA, there are no post translational modifications which are involved in protein and make the sequencing of protein difficult as compared to DNA. So, that was our topic today about protein sequencing. We have discussed the definition, we have discussed the methods involved, we have discussed the steps of protein sequencing, and we also understood why protein sequencing is important, why it should be done, and what are the challenges